Chapter 6 deals with genetic strategies and therapeutics. Just as a brief review of what we do in Kaplan Medical regarding molecular biology, there are 7 chapters in our book in molecular biology. The first 4 chapters of the 7 dealt with basic fundamental molecular biology. Chapter 1 dealt with DNA versus RNA structure and the issue of chromosomes and what they are about. Chapter 2 deals with DNA replication, that's DNA synthesis. Chapter 3 dealt with transcription, that's RNA synthesis. Chapter 4 dealt with translation, that's protein synthesis. And Chapter 5 was regulation of eukaryotic gene expression, a very high yield chapter for a national board, because that dealt with how we integrate basic molecular biology with the other disciplines. Virtually all disciplines is in Chapter 5. Physiology, pathology, pharmacology is all the integration of how you turn on genes in eukaryotes. Now, Chapter 6 and 7, the remaining chapters in molecular biology, is the more modern molecular biology. Chapter 7 deals with techniques of genetic analysis, of how we can do genetic testing of diseases, how we can do DNA testing for person identification in forensics. So in that Chapter 7, we talk about and review RFLP analysis, PCR reactions, northern blots, southern blots, a very high yield chapter in ultra-modern molecular biology. Now, Chapter 6, which is this chapter, deals with genetic strategies and therapeutics. In the learning objectives of Chapter 6, we will identify steps in cloning recombinant DNA. We'll talk about what cloning is about and what recombinant DNA means. We'll also discuss and review understanding the major points of the Human Genome Project. The Human Genome was sequenced and completed in 2003. We'll talk about what that was about and the significance in medicine of the Human Genome Project. We'll also define DNA libraries. Now, there are 2 kinds of DNA libraries we'll review in Chapter 6. We'll talk about genomic DNA libraries. We'll talk about what that means. The other one is cDNA libraries. We'll talk about the advantages and disadvantages of each of these, the genomic DNA library versus the cDNA library. And lastly, another major learning objective is determine the application of gene cloning. Once we get a gene in large amounts, what can we do with it? We'll talk about how the gene can be used to prepare recombinant proteins that you as a physician will prescribe in treating diseases. We'll also talk about a human gene that has been cloned to conduct gene replacement therapy. So these are the learning objectives of Chapter 6. Recombinant DNA technology allows the DNA fragment from any source, any biological source, to be joined in vitro in a test tube with nucleic acid vector that can be replicated autonomously. In other words, let me translate that sentence. What it says is you take a DNA fragment like a gene, a human gene, and splice it with some sort of vector. And usually the vector will be some sort of bacterial vector. And that is recombinant DNA. Recombinant DNA is a splicing of DNA from 2 sources. And for national board exams, the 2 sources usually, doesn't have to be, but usually is some human gene, which is DNA, human gene, and splice it with some bacterial vector, which is also DNA. So you're splicing DNA from 2 sources, that's recombinant DNA. And for boards, it will be a human gene with some bacterial vector. But in reality, recombinant DNA could be a chimpanzee gene spliced in with some yeast vector. But for the boards, it will be a human gene spliced in with some bacterial vector. Now, why we want to do recombinant DNA technology? It's because this provides a means of analyzing and altering genes and proteins, to analyze genes which have a mutation in them. And it provides a source of a specific protein, such as recombinant human insulin, in almost unlimited quantities. In the past, 10 years ago, 15 years ago, in the treatment of type 1 diabetes, where did the insulin come from to treat type 1 diabetes? Well, the insulin came from sacrificing about a half a dozen pigs or a half a dozen sheep to get enough insulin to treat human type 1 diabetes. But nowadays, that insulin used to treat type 1 diabetes, it is human insulin that was generated by recombinant DNA technology. And then recombinant DNA technology can also be used to provide reagents for genetic testing and prenatal diagnosing of genetic diseases. Now, the idea there provides reagents, and that's DNA probes that you'll discuss in chapter 7, DNA probes to conduct RFLP analysis, to do dot plots. DNA probes are needed for them, and recombinant DNA technology provides those probes. But in addition, recombinant DNA technology provides the genes to conduct gene therapy. So these therapeutic genes, which are good genes, used to treat an individual who has one gene that has a mutation in it, a missense mutation, a nonsense mutation. But if somebody has a mutant gene, we're trying to replace the mutant gene with a good therapeutic gene. And where did the therapeutic gene come from? Recombinant DNA technology. So recombinant DNA technology is a very powerful concept. That's the basis of this chapter. This slide is an overview of the cloning recombinant DNA process. What cloning means is where you have an original gene and you make millions of copies of it. As an example here or analogy, I should say, suppose we want to Xerox a particular document. You want to make large amounts of the document. First of all, you got to have the original document, and then you make millions of copies of it by putting the document in a particular machine. That's the same principle here of cloning of a gene. You have to have the original gene, and you make millions of copies of it. As an example, suppose I want to clone the Turco insulin gene. I want to make millions of copies of the insulin gene that came from me. Well, the first thing is, where do I get the original insulin gene? There are two sources of an original gene. One source is genomic DNA. I could do a biopsy of any cell in my body. If I want to clone the insulin gene, I don't have to go to the pancreas. I could, but I can just do a brain biopsy. If I do a brain biopsy and took the brain biopsy and put it in a test tube, in the test tube will be my insulin genes. One that I inherited from my mother and an insulin gene I inherited from my father. That's called genomic DNA in a test tube. Now, the other source of a gene is something called cDNA. Now, later on in the chapter, we'll talk about cDNA and how it compares to genomic DNA. But in this particular figure, I'm going to talk about genomic DNA as a source of a particular gene. Now, in that test tube, initially, that gene of interest is very tiny amounts of it, because it came from a biopsy, and it's very heterogeneous because there are other genes in that test tube. There are genes like the alpha globin gene, beta globin gene in that test tube. But if in this case here, I want to clone the insulin gene, it's very tiny amounts and it's very heterogeneous. And the goal is to amplify that piece of DNA. In this example was the insulin gene. The goal is to amplify it and make millions of copies of it. All right, now in that test tube of the genomic DNA, chromosomal DNA, what's done to start the cloning process, you cut up the DNA with a class of enzymes called endonucleases, which is coming up later on in the chapter, to generate many pieces of chromosomal DNA. And some of the pieces will have the insulin gene. Then we take those pieces of DNA that came from the chromosomes and splice it with some bacterial vector. It doesn't have to be bacterial vectors. It can be other kind of vectors. But by doing so, we generate recombinant plasmids. In this example, we have human DNA, human genes spliced in with bacterial plasmids, generate recombinant plasmids. And then we take those recombinant plasmids and give them to E. coli or any bacterium. Now, E. coli loves DNA. And so if you give bacterial plasmids to E. coli avidly takes up the bacterial plasmids And importantly here, that we have to generate a mixture of bacteria, each containing different recombinant plasmids. Some bacteria will have the plasmid which has the insulin gene. Other bacteria in this mixture will have plasmid which has the alpha globin gene. Other bacteria have the beta globin gene or whatever. So we have a mixture of bacteria containing different recombinant plasmids. Now, in microbiology, all of you have done this. You can take a mixture of bacteria and streak the bacteria on soft agar dishes. And each bacterium will sit down in an agar. You come back the next day, and each bacteria would form a colony on a particular dish that has agar. Each bacterial colony harbors a different recombinant plasmid. So one colony came from a single cell. That colony might have the insulin gene. Another colony may have the alpha globin gene. Another colony may have the beta globin gene. And importantly, on that dish of agar, on all these colonies, the total colonies on that dish contain all of the genomic DNA or cDNA that came from the top of the figure as the source of the gene, the source of the DNA. But on that plate, on that dish, the total colonies contain all of the genomic DNA, and that's what's referred to as a DNA library. Either a genomic DNA library or a cDNA library. One of the colonies will have the alpha globin gene. Another colony will have bacteria that has the insulin gene, and so on. Now, once we identify which colony has the insulin gene, now don't worry about how do you identify which colony has which gene. That's stuff you don't have to know for the boards. But once you identify which colony has a gene of interest, in this example, we're talking about the insulin gene. Now, suppose shown here in the purple, suppose that's a colony that came from one cell that has the insulin gene. And by using a toothpick or a tip of a spatula, we can dig up that colony, or theoretically, all you need is one cell from the colony. You take that one cell and you put it into a test tube or a flask, which has growth media. You can take that cell that came from the colony and now grow that cell. That bacteria will continue to grow and grow and grow. We have now successfully cloned the insulin gene in this example. Whereas as the bacteria grows and replicates itself, the bacteria is replicating the plasmid, which has the insulin gene in it. We have cloned the insulin gene. Now, what can we do with the cloned insulin gene? One thing is we can take the bacteria and turn on the gene of interest to now make recombinant proteins. As the bacteria grows, it's now making, in this example, making large amounts of the insulin protein. It's called a recombinant insulin protein. And then obviously that can be used to treat type 1 diabetes. Now suppose we don't want the insulin protein. We want the insulin gene. Well, what can be done, therefore, is to take the bacteria that now grows in large amounts. We can now chemically lyse the bacteria with detergents that you don't have to know. And we have now generated large amounts of the insulin gene. The DNA for insulin has now been amplified. And we have cloned the insulin gene. Notice here we have now generated large amounts of it, large amounts of the insulin gene, and it's now homogeneous. Homogeneous meaning there is no other human genes present. Large amounts of it and homogeneous. And what can you do with a cloned gene? Two things you can do with a cloned gene. One is you can conduct a gene replacement therapy. Somebody has a mutant gene, and you're now trying to replace the mutant gene with a therapeutic gene that was generated by this page. The other thing that can be done with a cloned gene is to generate transgenic animals. Pharmaceutical companies use recombinant DNA technology to generate transgenic animals. But in this chapter, we're going to spend most of the time talking about gene replacement therapy of the use of a cloned gene.